Good morning, everyone. My name is Zuza Bieniawska, and I am heading Genomics Customer Support Team at Cooper Genomics. Today, I would like to tell you about the journey of your sample from the enrollment process through the lab up to the reporting stage. I would like briefly to start from discussing our forms because every case has to be submitted with the paperwork. Then I would like to take you through the enrollment process for PGT-A, PGT-M, and PGT-SR cases. And then I would like to briefly tell you about what happens to your sample when it gets to our lab. So how is it received, how is it processed, and how is it reported? Okay. So one of the forms that is required with all PGT cases is test requisition form. This form we kindly ask customers to send prior we receive samples for testing. With the test requisition form, we are capturing basic patient information, patient demographics. So we need their name, surname, date of birth, and this is the same for patient and partner. We also ask for the internal clinic ID numbers for your patients. And this is because we are using this information, those numbers, when communicating with you about the case. So we do not use normally names and surnames of the patients when I'm discussing cases with you. So apart from the patient information, what we need also to be captured is the specimen information. We look at the egg retrieval date, and this helps us to know when to expect a given case in our laboratory for testing. We also need to know what sample type we are going to receive, saying that pretty much all the cases are now TRF and biopsies. As test requisition form is the same for all our cases, there is a field about the test information. So we need to know what we are to test for. Is it PGT-A case, PGT-M, or PGT-SR? As you can see, all the tests, they've got two options. We can report a case with mosaicism or without mosaicism. With mosaicism reporting is our default option. PGT-M cases and PGT-SR cases, they have to be reviewed and approved before we can accept biopsies for testing. And that's what I'm going to tell you about on the next slides. We also need to capture very basic information about your IVF center. So where to send the results and who is the contact person at your clinic. Another form that is needed is the biopsy worksheet. Biopsy worksheet is to be submitted together with the samples. Once again, we need to capture patient information and some information about the sample and your center. So we need to know what we are testing for, so what type of test it is. We need to know the sample type, and we need to know if we are to analyze this case now, or are we going to batch it? So store it until further testing is required. Batching means for us that you can send us the samples. We would perform the very first step in sample processing that is called whole genome amplification. So we would amplify DNA from your cells, and we would store this DNA until further analysis is required. We proceed with the first step in sample processing. That's because DNA is much more stable during storage. We then process the case once we've got go-ahead from the clinic. We need to, of course, receive the information from what clinic we are receiving the case, biopsy date. This is the information that appears on the report. And we need to, of course, capture information about the biopsy samples received to us. So there is a field to put the name of the samples, and this name of the sample has to correspond with what you write on the top of the tube submitted to us for testing. Couple of other information about the sample. So how many cells were biopsied roughly? Of course, we know it's difficult to give the precise number of cells submitted to us for testing. Sample stage, embryo grade, And then who performed the biopsy? Once again, embryologist performing biopsy. This is the field that also appears on the report. In the square here, we are asking clinics to put the barcode from the biopsy kit. Every biopsy kit that is sent to you has got unique barcodes inside. So biopsy form, biopsy worksheet is to be submitted together with samples when they arrive to us for testing. There is one more form that is called enrollment form. This one is only required for PGT-SR and PGT-M cases. And with this form, we are capturing patients' contact details. And this is because for PGT-M and PGT-SR cases, we are organizing for a GC, genetic counseling enrollment call, between our GC team and the couple. And we need this information in order to set up a genetic counseling appointment. So what is the actual process to enroll the case with us? So let's start with PGT-A. So for PGT-A, the process is really straightforward. All that we need to receive from you is the test requisition form, and ideally would like to receive this one prior to the sample's arrival. We want to make sure that we've got enough time to set your patient, set the couple in our database, in our patient database. Please send this form to globalsupport@coopergenomics.com. Then we need to receive, of course, the biopsy form together with samples. And that's pretty much it. So the testing, PGT-A testing and reporting takes 2 weeks. We do often release those reports ahead of our quoted turnaround time. Okay, so let's now move into the PGT-SR case enrollment process. So for this one, we need to receive the karyotype report for patient and or partner, depending who is carrying translocation. And we need this report to start the whole enrollment process. We ask to send the karyotype report to our coordinator. Our lab team would carefully revise translocation itself and the breakpoints associated with it. If the case can be accepted, then our coordinator will provide you with translocation inquiry report. Accepting a case, accepting a PGT-SR case, usually comes down to the type of translocation and breakpoints, so the sizes of translocation. We just need to be confident that our platform will detect those. So if the case has been accepted and you received from us translocation inquiry report, then the next step is our coordinator will ask you to fill in enrollment form. And this is because we want to set up GC enrollment call between our team, our GC team, and your patients. After the call, our GC would send the short summary notes from the discussion with the patient, and we will be sending those to the clinic and to the patient. For PGT-SR cases, we need to receive signed PGT consent, and the consent needs to be signed by the couple. So once we've got the signed consent in place, we are ready to accept samples for processing. And then PGT-SR testing and reporting takes 2 weeks. We are often able to release this report ahead of quoted 2 weeks turnaround time. And the next type of cases that we accept, those are the PGT-M cases. So for this one, to start the whole enrollment process, we need to receive mutation reports for patient and or partner. And if there are any family members, they were tested and there are mutation reports available, we'd love to see those as well. So please send all the mutation reports to our coordinator. Then the GC team and the lab team will revise those documents. They may come back with more questions or they may request further documentation. At this point, our coordinator will check if there are any other family members available for DNA sample donation for the test design. And this is the way our PGT-M platform works, because in order to assess presence or absence of a specific allele in embryos, we need information, we need the DNA information from the couple and, if possible, from references. If there are no other family members apart from couple there to give DNA for testing, there may be other possible PGT-M protocols or designs, but this is something that would be discussed further with the GC and the lab team. So once the case has been accepted, we will ask to fill in the enrollment form, because we want to set up GC enrollment call between patient and the GC team. So once again, after the consult took place, our clinic, our coordinator will be sending a GC summary note to the clinic and to the patient. Again, we need to receive a PGT consent that is signed by the couple. Briefly, I would like to mention what happens during the GC enrollment calls, and this is for both for PGT-M and PGT-SR cases. So the aim of those discussions between our genetic counseling team and the couple is to discuss the specific PGT-M or PGT-SR case, what to expect from the results, what to expect from testing, what is the accuracy of testing. If patient has got any questions about PGT-M or PGT-SR, those can be answered by our GC team. So after the genetic enrollment call, the next step would be to dispatch cheek swab collection kits. So this is the most common way how we collect DNA from the couple and from the family members. So we send those by post or by courier. This really depends on the location. And this is the very easy and straightforward kit to be used. There are a lot of instructions together with the kit. You just need to simply take a DNA sample from the inside of your cheeks and send it back to us. In some of the cases, if there is already DNA extracted, we can accept those as well. What we cannot accept is the blood sample. So the next step is to start the assay optimization. So once all the cheek swabs with the collected DNA are received in our lab, we start with the case workup. This takes usually up to 4 weeks. And every PGT-M test has the bespoke design. So it is designed for a couple. Case optimization, once the case optimization is finished, our coordinator would notify the clinic that trophectoden and biopsies for testing can now be received. And it is only then that we can really accept samples for testing. So the whole case enrollment process, from the moment that we receive mutation reports up to the stage that we notify you that the case optimization has been finished, usually takes minimum of 8 weeks. And this is because we need time to revise documentation submitted to us. We need to have time to organize for the GC enrollment call. And then we need to receive all the DNA, and the whole optimization takes 4 weeks. That's why usual case enrollment turnaround time is minimum of 8 weeks. PGT-M testing and reporting takes 2 weeks. Okay, so now your PGT-A sample or PGT-M or PGT-SR case has been completed in your laboratory and it's ready for shipment to us. So we would kindly ask that if you have already the courier tracking number for your parcel available, please email this to global support. Let us know how many boxes and how many cases we are expecting. And this is because once the case arrives to our London laboratory for testing, our team, global support team, will send you confirmation on the case arrival that the case got to us safely. So when the case arrives to the lab, it lands in the dedicated area for the sample reception. So, and there are several checks carried by the lab team on the case arrival. Sample reception is usually carried out throughout the day, and there is a dedicated lab team members. From our team, from the global support team, there is a dedicated support member who will acknowledge the case to you on arrival. So when the parcel gets to our testing lab, it is first inspected from outside, and then it is opened. We take out all the biopsy boxes, and then we visually inspect every box from outside, and inside we inspect each tube. So we check, is this the case called on arrival? Then we take out every tube and we look at the volume of the samples. We do not measure the volume, but we know we are expecting around 2.5 microliter of a buffer in the tube together with the cell. We then check all the names of the tubes on the top, from the top of the tubes, and we also check all the barcodes if they are matching the paperwork. The next check that we are carrying out is really to compare the biopsy box that arrived to us. So on the top of the biopsy box, there is a field to put the patient identifiers, and we check this biopsy box versus test requisition form submitted and the biopsy form. If everything is all right, then we record the case on our dedicated trackers. And now it's ready for processing. So for PGT-A and PGT-SR cases, samples, this is how the sample processing looks like. So we start with the whole genome amplification. So we want to amplify from the couple of cells that you submitted to us, we want to amplify the DNA. Once the DNA is amplified, we'll check results of this reaction on the agarose gel. After that, we are preparing libraries, and those libraries are later on being sequenced. We are using next generation sequencing platform. Data is analyzed via PGT-A technology platform. Once the results are ready, they are sent in a password-protected document to the email that was given by your clinic to us. And if there are any questions about the results or reports, please send them to the global support team so we can make sure that they are answered as quick as possible. When it comes to the PGT-M testing, we are using carrier mapping platform. This is bit array based technology, and carrier mapping uses biomarkers or SNPs within the genome. So this platform enables screening of embryos for likelihood of carrying defective gene. Where possible, and this really depends on the mutation itself, on the condition, we would be also applying sequencing. And we also perform next generation sequencing, so NGS for PGT-A part of testing. So we run PGT-M part of testing first, and all the embryos from PGT-M testing that are found to be suitable for transfer would be then analyzed on PGT-A to report ploidy status. So we not only report on the allele gene or mutation inherited status, but also if the embryo is euploid or aneuploid. So the key steps in sample processing is, once again, we have to do the whole genome amplification to get DNA amplified from the cells that you submit to us. We prepare libraries that are later on analyzed, and we analyze the data. So password protected report will be sent to the clinic once it is ready. And again, if there are any questions about the results and reports, please send them directly to global support. All the reports will be emailed directly by the lab team to your clinic, but if there are any questions, please send them back to the global support team. I hope that you enjoyed the story about the journey of the sample from the enrollment process through the lab up to the reporting. And if you've got any questions, please call us on this number or email us your questions to globalsupport@cupergenomics.com. Thank you very much.